Hello, General Biology, and welcome to the fourth in the screencast series of Chapter 8, Photosynthesis. And what you are looking at is the electron transport chain. And remember when we were talking about that in the last screencast, this electron transport chain is found within the thylakoid membranes and is where the light-dependent reactions are going to take place. So just to orient ourselves here, and I'm going to try to work with the pen a little bit, this is going to be the lumen, the inside of the thylakoid membrane. It's a little bit hard to write with. I'm hoping you can see that. This part right here is going to be the stroma. And again, this is the thylakoid membrane. I'm not going to write that part. All right. Let's familiarize ourselves with the structures. I'm going to take off the pen. The structures that we have embedded in the membrane. This is photosystem 2, and this is photosystem 1. The smaller blue dots are transport proteins that are embedded in the membrane, and this larger blue dot is an ion pump. And then this last orange or reddish orange is ATP synthase. Again, it's a special type of transport protein, and it's going to work by facilitated diffusion, where the ion pump's going to require energy in order to work. All right. Notice here inside the lumen, we have a high concentration of hydrogen ions, where outside we only have a small concentration of hydrogen ions, and that's going to be significant. Notice also in the lumen we have water molecules, and we need that water molecule because it's going to give us hydrogen ions, it's going to give us electrons, and it's going to give us oxygen. The oxygen we're not too concerned about because that's going to leave the cycle, but we're going to watch what happens with the hydrogen ions and with the electrons. So the process starts with photosystem 2, and you can see this is light energy. Light energy is going to get trapped in photosystem 2, which has the pigment chlorophyll. And that light energy, we talked about it with a driveway. When you have light energy on a driveway, it heats up and it gets warm. Well, that's kind of what's going to happen here. It's going to heat up, or what they call excite, excite the chlorophyll. And when it does that, any water that happens to be in its presence is going to wind up splitting, where you'll get the hydrogen ions and you'll get electrons. The hydrogen ions are going to collect within the lumen. The oxygen, as I said, is going to leave the process, and the electrons are going to stay within the photosystem. So being pulled away, being ripped away from the electrons, the hydrogen ions, the electrons require a little bit of energy, so that light energy coming back into photosystem 2 is going to re-energize those electrons. Okay, so once they've been recharged, the electrons are going to move into the first transport protein. Nothing's going to happen here. It's more like a stepping stone. It's a way of getting electrons from photosystem 2 to the ion pump. So they'll move to the first transport protein and then into the ion pump. When the electrons get into the ion pump, they're kind of like the battery. Plug it in and that pump is now working, and that pump will move hydrogen ions against the gradient from a low concentration to a high concentration into that inner thylakoid space, that lumen. Once the electrons are done pumping the hydrogens, they'll move to the next transport protein. And again, this is just like a stepping stone. It's a way of getting the electrons from the ion pump into photosystem 1. Now, Let's say you use your cell phone, and you've used your cell phone. You started out with 4 bars on your battery, and now you're only down to 1 bar. What do you usually do? Recharge it, right? Okay, so these electrons are down to 1 bar. They need to be recharged. So in photosystem 1, that's what's going to happen. The energy from sunlight is going to be trapped in the chlorophyll of photosystem 1. And again, they're going to get excited to the point where they will re-energize the electrons. And the re-energized electrons will then move to the last transport protein, which is kind of like a bus station. And they'll wait until they can be picked up by an electron carrier. There's electron carriers called NADP+ that are present within the stroma. Remember, we called it a suspension. So they are present within that suspension. And what's going to happen is the negatively charged electrons are going to be attracted to these positively charged electron carriers, as well as the hydrogen that they used to be attached to, and they're going to join together with NADP+, and they're going to form the energy carrier NADPH. And this NADPH is going to go off to be used in the Calvin cycle. So now, as a result of this electron transport chain, it's created a high concentration of hydrogen ions in that lumen. So what's going to happen, from the natural forces of diffusion, these hydrogen ions are going to move from an area of high concentration to an area of low concentration. And they're going to do that through this special transport protein called ATP synthase. ATP synthase is like a protein channel. So the hydrogen ions will just pass through. But when they do, they're going to cause that ATP synthase to spin. Just like I said earlier, just like a stirring action. And as it stirs, that means that the substances that are inside of the stroma are going to be pulled together. So ADP, along with some independent phosphate groups, will be stirred together by this ATP synthase to make ATP. And again, that ATP will go off to the Calvin cycle to be used, or the light independent reactions to be used there.